Hello, everybody, and welcome back to this discussion on strategies for gene cloning. So we've discussed the isolation of a single gene from a complex source utilizing genomic libraries. Now I want to discuss cDNA libraries, so let's delve into that. Now, a cDNA library is generated by reverse transcription of cellular messenger RNA, beginning with the first strand DNA synthesis on the messenger RNA template, followed by the second strand DNA synthesis. What you have to recognize here is that in a cDNA library, because you are reverse transcribing cellular messenger RNA, you lack introns, which may be located within the coding sequence itself or in the 5' or 3' untranslated regions. Now, irrespective of their location, introns are copied by RNA polymerase, followed by RNA processing, which removes those introns. This is a process known as splicing, which does not occur in bacteria, and thus eukaryotic cDNA cloning becomes important when endeavoring to express a eukaryotic gene in a bacterial host. Introns do exist, although rarely in bacteria, so keep that in mind. They do exist in bacteria, although this is very rare. Here, it's important to recognize about reverse transcription of cellular messenger RNA. With regards to that, it's important to recognize that the cDNA library is representative of the RNA population from which it is derived, and thus it's dependent on the cell type, and it's dependent on the developmental stage. And so contents of a cDNA library will vary in accordance with such parameters. And in addition, the contents will include alternative splice variants of differentially spliced genes. So you have to keep those things in mind. There are various methodologies by which cDNA libraries are generated. There is the Manietis method, so let me discuss that first. So the Manietis method for the generation of cDNA libraries involves utilization of oligo-DT primer annealing. This facilitates first-strand cDNA synthesis, and after which the newly synthesized strand will fold back on itself to form a hairpin loop. This results in self-priming of the second strand, after which the hairpin loop is cleaved with an S1 nuclease. Recall the S1 nuclease is an endonuclease that cleaves single-stranded DNA, and this permits insertion into a cloning vector. Now, what you have to recognize here is that the oligo-DT primer anneals to the poly A tail, and this facilitates the first strand cDNA synthesis. This is synthesized by reverse transcriptase, obviously. And what you ought to recognize here is that self-priming of the second strand synthesis happens via DNA polymerase. So we utilize a DNA polymerase for the synthesis of the second strand, whereas reverse transcriptase for the synthesis of the first strand of cDNA. Now, a disadvantage of the hairpin loop being cleaved with the S1 nuclease is that you can have partial loss of the sequence at the 5' end, so you have to keep that in mind. There's also a drawback when using oligo-DT primers in reverse transcription, and that is 3' end bias. So 3' end bias translates into a preference for clones representing the 3' end of cDNA. So you have to recognize that using an oligo-DT primer results in 3' end bias, which translates into a preference for representing the 3' end of cDNA. This is mitigated by utilizing random primers in the first and second strand synthesis of cDNA, usually utilizing hexamer oligonucleotides. Yet that results in much smaller clones, and in turn, cDNA must be assembled from shorter fragments. Yet as cDNA increases in size, difficulty in isolating full-length clones ensues. So you have to keep those things in mind. So I said that there can be difficulty in isolating full-length clones that ensues as a result of this, and there are several approaches to resolving this that have been delineated. So we can select messenger RNA with intact 5' ends. An example methodology of this would be that by EDRI, which involves oligo-DT primer annealing, facilitating the first strand cDNA synthesis. Again, the oligo-DT anneals to the poly A tail, and then this is followed by treatment with RNase A. You want to keep in mind here that the full-length cDNAs will reach the 5' end cap, and on the other hand, the partial-length cDNAs will not reach the 5' end cap. So when you treat with RNase A here, this results in degradation of single-stranded RNA, and thus DNA-RNA hybrids remain intact. Now, the full-length cDNAs, as I said, will reach the 5' end cap and are therefore protected from cleavage by RNase A. On the other hand, the partial-length cDNAs will not reach the 5' end cap and thus are therefore digested in the single-stranded region by RNase A. Following treatment with RNase A, the eukaryotic initiation factor 4,4E is utilized to isolate full-length cDNAs, and this is performed via affinity capture or column chromatography, and thus eluding the full-length cDNA. Now, more specifically, this eukaryotic initiation factor will bind to the 5' end cap, which is only located on the full-length cDNAs. There is another approach to resolving the issue that has been delineated, and that is the selection of the messenger RNA with intact 5' ends using oligocapping. So this involves treatment with alkaline phosphatase. In turn, this removes the phosphate groups in the 5' ends of uncapped RNA. And in turn, this has no effect on the 5' capped RNA. Now, this is followed by treatment with acid pyrophosphatase. In turn, this removes the cap from full-length RNA, and in turn, leaving the 5' terminal phosphate group. This is followed by capping the full-length RNA with an oligonucleotide, thus ending up with an oligocapped population of RNAs. And then this is followed by reverse transcription using an oligo-DT primer, which is then followed by second-strand cDNA synthesis with PCR using an oligo-DT primer and primer annealing to the oligonucleotide cap.